I think we can continue with the biochemistry chapter 7. So, the biochemistry chapter 7 is about the RNA and the genetic code. And let's start with the genetic code. So, we have central dogma, which is basically when the DNA is being transcribed into the RNA, and then the RNA is translated into the protein. We have degenerate codes that allow for multiple codons to encode for the same amino acid. I think this kind of relates to wobble that we have here as well. So just keep in mind that we have degenerate code. The whole concept of degenerate code is the fact that you can code for the same amino acid with multiple codons. Then we have start and stop codons. So we have initiation, which is AUG for start codon, and we have termination for stop codon. So how I memorize the stop codon is that first we have yuga, so yoga basically, like UGA, and then just reverse it. It's UAG, and just get rid of the G and replicate it with A, so it's UAA, right? There you go. We have it, Yuga, and then reverse it, UAG, and then just replicate A, so it's UAA. The wobble is basically the third base in the codon that allows for mutations to occur without effects in the protein. So you can change the wobble and still you'll get the same protein. Wobble base pairings are less stable. That's also something that we need to know. I did not know that. Point mutations could be silent, so mutations with no effect on protein synthesis, just like the wobble. That's a silent mutation that's usually found in the third base of a codon. We have nonsense truncation. The truncation is basically non-stop. You can memorize it with non-stop. So NON, nonsense, is for when the mutation produces a premature stop codon. The missense, also called truncation, right? Nonsense. The missense is a type of mutation that produces a codon that codes for a different amino acid. So the missense is the mutation that produces a codon which codes for a different amino acid. We have the frame shift mutation. It results from a nucleotide addition or deletion and changes the reading frame of subsequent codons. So the frame shift is when the nucleotide addition or deletion changes the reading frame of subsequent codons. And we have RNA. It's similar to DNA, except it's a ribonucleic acid, not a deoxy. So the ribose sugar instead of deoxyribose is also uracil instead of thymine in the RNA, and it's single-stranded instead of double-stranded. The three types of RNA, however, we do still have double-stranded RNAs for the viruses. But as far as we're talking about our body, we have single-stranded RNAs. The three types of RNAs are messenger RNA, mRNA. They are transcribed from DNA in the nucleus. It travels into the cytoplasm for translation. So it's transcribed in nucleus and it's translated in cytoplasm. Transfer RNA, tRNA, it brings in amino acids and recognizes the codon on the mRNA using the anticodon. So we have mRNA, and also let's just finish with rRNA, ribosomal RNA. It makes up the ribosome and is enzymatically active. So the feature, what's the feature about mRNA, the messenger RNA? The messenger RNA is transcribed in the nucleus and translated in the cytoplasm, right? The tRNA, brings in the is it the amino acids? Yes, it brings in the amino acids and recognizes the codons as codes for the recognize the codes on the mRNA using its anticodon. So it's the tRNA that has anticodon. And the rRNA, ribosomal RNA, has enzymatic activity, right? It's enzymatically active, and it makes up the ribosome. So ribosome by itself is a protein, right? Therefore, the ribosomal RNA is translated and makes up the protein. Now transcription. We can see the appendix for the full diagram, but for transcription we have helicase, which unwinds the DNA double helix. So that's for helicase. We have RNA polymerase 2, which binds to the TATA box within the promoter region of the gene. 25 base pairs upstream from the first transcribed base. RNA polymerase 2 binds to TATA box within the promoter region of the gene. 25 base pairs upstream from first transcribed base. Upstream. hnRNA, collective term for the unprocessed mRNAs in nucleus. So when the mRNA is not processed, it's in the nucleus. We call this the hnRNA. We have post-transcriptional modification. Now, keep in mind we have post-translational modifications here, and we have the post-transcriptional modifications. These are two different processes. So the process in eukaryotic cells where primary transcript RNA is converted into mature RNA, introns cut out. That's for post-transcriptional. So exons exit the nucleus and form mRNA. Introns spliced out so they stay in nucleus. Introns also enable alternative splicing. So introns enable alternative splicing. Alternative splicing, what is it? Usually introns are cut away and exons remain, but alternative splicing might change that. A certain exon may be cut out or an intron may stay. This allows for the RNA segment to code for more than one gene. Alternative splicing. So the effect of alternative splicing, sometimes the intron staying in, is that it allows for the RNA to code for more than one gene. 5' cap also and poly tail are added to the mRNA. So 5' cap and poly tail are also the second and third modifications for post-transcription. The cap and tail stabilize mRNA for translation. Prokaryotic cells can increase the variability of gene products from one transcript, though, through, polycistronic genes. Should know what these are and figure out what they do. There are multiple translation sites within the gene, which leads to different gene products. So multiple translation sites within the gene, which leads to different gene products. Multiple translation sites within the gene, which leads to different gene products. Different gene products. Cool. We can also make this green. Cool. Next one. For translation, we have tRNA. It translates the codon into the correct amino acid. Yes, the ribosomes factories, they are factories where translation protein synthesis occurs. So yes, we know that translation happens in ribosomes. For eukaryotes, they are 80S ribosomes, and for prokaryotes, it's 70S. It's more always for the eukaryotes. We have initiation. So prokaryotes is when the 30S ribosome attaches to the Shine-Dalgarno sequence and scans for a start codon. It lays down N-formylmethionine in the P side of the ribosome. For eukaryotes, when the 40S ribosome attaches to the 5' cap and scans for a start codon, it lays down methionine in the P side of the ribosome. So the 5' cap is the unique feature of the eukaryotes. For the prokaryotes, it's the Shine-Dalgarno sequence. We have also elongation, the addition of a new amino acid tRNA into the A side of the ribosome, and transfer of the growing polypeptide chain form the tRNA in the P side from the tRNA in the P side to the tRNA in the A side. So if you don't know, we have the APE. So it arrives on the A side of the ribosome, the elongation happens in the P side, and the E side is for the exits, right? So in the P side, the tRNA is bringing all these amino acids and attaching them to the polypeptide chain that is elongating, and when it's finished, it exits from the E side. So the elongation again, the addition of a new amino acid tRNA into the A side of the ribosome, and transfer of the growing polypeptide chain from the tRNA in the P side to the tRNA in the A side. The now uncharged tRNA pauses in the E side before exiting the ribosome. It's uncharged when it wants to exit. The A side tRNA moves to the P side. And termination occurs when the codon in the A site is a stop codon. Release factor places a water molecule on the polypeptide chain and thus releases the protein. We have RF, release factor, replacing a water molecule on the polypeptide chain and thus releases the protein. Then we have post-translational modifications. They fold by folding by chaperones. So we have protein folding and unfolding. And when we are folding the protein, you are when you're unfolding it, you are making it ready for translation. But when you are folding it, you're just saying it's enough. We have translated the protein. Now let's get over with this. And the folding by chaperones is what takes place in the post-translational modifications. The formation of quaternary structure is also that in post-translational modification. Cleavage of proteins or signal sequences. Covalent addition of other biomolecules like phosphorylation, carboxylation, glycosylation, prenylation. We don't know what prenylation is. Might be the addition of a fat. And at the end we have DNA ligase, which fuses the DNA strands together to create one strand. So fusing the DNA strands together to create one strand. The control of gene expression in prokaryotes. The Jacob-Monod model ligase is attaching, fusing the DNA strands together to create one strand. The control of gene expression in prokaryotes. Jacob-Monod model explains how operons work. What are operons? They are inducible or repressible clusters of genes transcribed as a single mRNA. Again, inducible or repressible, you can promote it or demote it. Repressible clusters of genes, they are a bunch of genes transcribed as a single mRNA. So they are transcribed as a single mRNA. Transcribed? Yes. They're transcribed as a single mRNA in order to be translated later. Inducible systems, they are under normal conditions. They are bonded to a repressor. They are turned on when an inducer pulls the repressor off. For example, lac operon. Then we also have repressible systems transcribed under normal conditions. They can be turned off by a co-repressor, coupling with the repressor, and the binding of this complex to the operator side. Example, TRP operon, is it tryptophan, maybe? And control of gene expression in eukaryotes, transcription factors, search for promoter and enhancer regions in the DNA, then bind to the DNA and recruit RNA polymerase. Promoters are within 25 base pairs of the transcription start site. I think we saw that here, right? Promoter region, 25 base pairs. Transcription factors, again, search for promoter and enhancer regions in the DNA, then bind to the DNA and recruit RNA polymerase 2 here, right? Then bind to the DNA after searching for promoter and enhancer regions. And the promoters are within 25 base pairs of the transcription site. Enhancers are more than 25 base pairs away from the transcription site, start site. Modification of chromatin structure affects the ability of transcriptional enzymes to access the DNA through histone acetylation. Increases accessibility, the histone acetylation, and DNA methylation decreases accessibility. Let's make these green. Let's make these pink. Cool. So again, modification of chromatin structure, this is in eukaryotes, affects the ability of, so eukaryotes, let's make it blue, and prokaryotes, let's make it purple. Modification of chromatin structure affects the ability of transcriptional enzymes to access the DNA through histone acetylation. So acetylation of the histone increases its accessibility, or DNA methylation that decreases the accessibility of the DNA. We assume that increasing accessibility, histone acetylation, enables the DNA to expand, right, to be available. But the DNA methylation, the methylation of these histones basically encloses, folds the DNA together, so you cannot transcribe it. Cool. So we did 16 minutes and a half on Biochemistry chapter 7. So just a quick recap. We learned about the the basically genetic code, the RNA, and the first few concepts were from, how DNA differs from the RNA, and then we went through transcription, the central dogma, and translation. Transcription has its own post-transcriptional modifications. It's the addition of the 5' cap, the poly A tail, the introns and the exons. We have alternative splicing that is enabled by the introns, and introns bring less stability, right? Here, introns bring less stability. Sorry, wobble base pairing actually brings less stability. Then we had non-stop, which is for stop codon mutation. Non-sense mutation. Missense is when you just mutate a different amino acid, right? Here, missense, different amino acid. When you have frame shift mutation, you have 3 types of RNA: the mRNA, which is transcribed in the nucleus and translated in the cytoplasm. You have tRNA bringing in the amino acids, and rRNA, ribosomal RNA, they have enzymatic activity. They are enzymatically active, and they make up the ribosome. So in the transcription, we talked about alternative splicing, the addition of poly A tail and stuff like that. Then in post-translational modifications, we do have the chaperones, unfolding the we have folding by chaperones, right? And formation of quaternary structure and cleavage of proteins or signal sequences. So you just cut out the proteins or signal sequences, and you have covalent addition of other biomolecules like phosphorylation, prenylation that we don't know still what it is, and carboxylation and glycosylation, and DNA ligase for fusing the DNA strands together to create one strand, right? And what else? Yeah. So now let's do physics chapter 9 and 8. The biology flashcards, I'll do them in a minute, but for this one, let's do physics chapter 9 and 8. Should be down here. So this is where we left off for, let me start my timer. So the alpha decay was the alpha for the, so we have an atomic number and an atomic weight. The atomic weight is the top number is A, the atomic mass, right, weight, whatever, and we have the one at the bottom, which is the atomic number, right? And for alpha decay is 4 plus 2, so whatever reactant you have, you'll have the alpha, so 4 plus 2, and then the other atom, which is the product, is minus 4 on the atomic weight and minus 2 on the atomic weight number. These are much easier in practice. So we have beta negative decay, so the decay of a neutron into a proton with emission of an electron. and an anti-neutrino. So electron, beta minus, and anti-neutrino. With beta positive, you get neutrino. So anti is negative, it's easy, beta negative. With gamma decay, you have the emission of a gamma ray made up of photons, so with beta, positive decay is also positron emission. In gamma decay, which converts a high energy nucleus into a more stable nucleus. And we have electron capture, which is the absorption of an electron from the inner shell that combines with a proton in the nucleus to form a neutron. So the absorption of an electron from the inner shell that combines with a proton in the nucleus to form a neutron. So absorption electron capture, absorption of electron from the inner shell that combines with a proton in a nucleus to form a neutron. Yeah, I don't know what this is, but so the electron comes to the reactant side, and as a product you get an element with the Z-1. Half-life, the amount of time required for half of a sample of radioactive nuclei to decay, or the time it takes to reduce the radioactivity of a substance by half. Exponential decay, the rate at which radioactive nuclei decay is proportional to the number of nuclei that remain. N equals n naught e to the negative lambda t. Lambda is known decay constant, and these are numbers. Note if the problem just says beta, they mean beta negative. Beta negative is the default. Beta negative is the default. So beta negative is the default, not beta positive. Cool. So let's go to the previous chapter here. We have electromagnetic spectrum. So we have electromagnetic waves that transfers. So this is about light and optics, right? Now, for electromagnetic waves, what does come to mind, right? Electromagnetic, electro from electrons, magnetic from the magnets, right? Diamagnetic and paramagnetic we had in the first chapter of chemistry. The paramagnetic being able to be attracted, pulled into a magnetic field. Diamagnetic because those two electrons are spinning in the opposite direction. They just neutralize each other, so you don't get any magnetic attraction. You are pushed out, you are repelled in the magnetic field. So back to electromagnetic waves. Yes, this comes to mind, right? The high frequency to the low frequency waves that we see, and these are transverse waves. Transverse, it doesn't mean that they are horizontal like this. Let's see. That consist of an oscillating electric field. So they have an oscillating electric field. Oscillating means like it's going back and forth, right? And an oscillating magnetic field. So they have both magnetic and electric fields, and these two fields are oscillating, going back and forth. In order to know more about these, we need to go back to the previous chapters and not the waves, actually the waves and sound here, right? Do we see transverse waves here? No, but might be somewhere in between. This is about the circuits. This one is about electrostatics and magnetism. Yeah, I think this is a good one to look at. Yeah, we can come back to these, but what I wanted to, this is about fluids. Everyone loves that. I'm joking. Thermodynamics here. energy and work, and yeah, kinematics and dynamics. So yeah, let's just continue. But all I wanted to mention was that these electromagnetic waves are basically based on the knowledge that we need to have from the electric field and the magnetic field. It's going to be in the electromagnetic chapter. The two fields are perpendicular to each other and to the direction of propagation of the wave. So perpendicular to both each other and direction of propagation of the wave, right? The electromagnetic spectrum, the range of frequencies and wavelengths found in the electromagnetic waves. We have electromagnetic spectrum, small lambda equals high energy. Yes, here, and high lambda low energy. So this is low energy, this is high energy, and it goes from radio waves to microwaves to infrared to the visible light, and then to ultraviolet to UV, right? X-ray, and then we have gamma rays. And the length is also good to know, like the frequencies or the wavelengths of the waves, right? Note gamma, X-ray, and higher UV are ionizing. Ionizing, what does that mean? Does it mean that they can make an ion or turn a molecule into an ion or atom to an ion? They can liberate electrons from nearby atoms and create free radicals. We had seen free radicals like the reactive species, like the oxygen, right? Superoxygen. It has only one electron on the atom, right? In the valence electron. They can liberate electrons from nearby atoms. So liberating electrons from nearby atoms. We can check that out later. We have the visible spectrum. It goes from 400 to 700 nanometers. The higher the wavelength, the less the energy and the lower the frequency, right? So the red is lower energy than the UV, the violet, or the purple, or even the blue, right? And we have hydrogen spectral series. What are these? Lyman, ultraviolet, N equals 1, Balmer, visible, N equals 2, Paschen, infrared, N equals 3, and acrostic loves beer pong, then N equals 1, 2, 3. What the hell are these? I have no clue. You can check that out later. Let me just note this down. Hydrogen spectral series. Hydrogen spectral series. Lyman, Paschen, Balmer. Lyman, Balmer, Paschen. Rydberg. And what is the Rydberg formula? It's HF, which is the same as the wavelength, right? I'm sorry, it's equal to C, the light speed, right? The energy, I think. Rydberg formula, HF equals R, big R, times the 1 to the N2 final minus the 1 to the N2 initial. We'll figure that out later. So Rydberg formula. Diffraction. What is diffraction? The bending and spreading out of light waves as they pass through a narrow slit. Diffraction may produce a large central light fringe surrounded by alternating light and dark fringes with the addition of a lens. Interference when waves interact with each other The displacements add together in a process called interference. When the waves interact with each other, the displacements add together in a process called interference. And diffraction is the bending and spreading out of light waves as they pass through a narrow slit. Diffraction may produce a large central light fringe surrounded by alternating light and dark fringes with the addition of a lens. Young's double slit experiment shows the constructive and destructive interference of waves that occur as light passes through parallel slits, resulting in minima, dark fringes, and maxima, bright fringes. Of intensity. So it's about interference, right? We have also polarization. We also have polarization. A polarizing filter only lets light through if the electric field of the wave aligns with the openings in the filter. The electric fields of the exiting light oscillate along the same axis. Circular. What did we just read? Plane polarized light. We know this is a plane polarized light demonstration. So direction of light waves oscillate in all directions. Direction of light propagation, light waves oscillate in a single plane polarized light. This is a polarizer. Circular polarized light, all of the light waves have electric fields with equal intensity but constantly rotating direction. Equal intensity but constantly rotating direction. Circularly polarized light is created by exposing unpolarized light to special pigments or filters. Intensity, rotating direction. Circularly polarized light is created by exposing unpolarized light to special pigments or filters. And so it's plane versus circular polarized light. A polarizing filter only lets light through if the electric field of the wave aligns with the openings in the filter. Only if E field aligns with openings in the filter. Now we have the geometric optics. Reflection is the rebounding of incident light waves at a medium's boundary. The law of reflection, theta 1 equals theta 2 incident ray, reflected ray, incident light waves. We have spherical mirrors, so mirror, concave, convex plane. We have the image produced in the concave is real. If it's inverted, if it's upright, it's virtual. The object's position is greater than the focal length, so it's here. You can see in the concave mirror, it's upright, it's virtual, and the object's position is less than the focal length. Now this is the center, this is the center of curvature, this is the focal point. Focal point is half of the center, and it's real if it's inverted. The opposite position is greater than the focal length. So it comes here, it goes here, it comes here, it goes here. So we get, if it's this way, you will have, if it's after the center, after focal point, basically, I think, is less than the focal length. It's greater than the focal length. Doesn't make sense still, since it should be here. So wait, if we have the object, this is the image, so it's greater in the center. It's saying like this, it goes through the focal point, goes here through the focal point, and this is the image you get. It's also inverted. Now if you place it here, you have to go here and go there. You have to go here and go there. That's how you get the upright virtual image less than focal length. Now for convex mirrors, you have virtual, still up, it's all virtual. Here you get upright and smaller, so it goes the head to the focal point, and you do another one through the center, right? That's how you get the image. For plane is virtual, upright, and same size, and you can think of these as spherical mirrors with infinite radii of curvature. We know how the plane images are, so let's do for convex, concave, green, real, inverted, and blue, virtual, upright, smaller. We also have something called refraction. So refraction is the bending of light as it passes from one medium to another. That's from water, for example, to oil or any other liquid. The speed of light changes depending on index of refraction of the medium. This speed change causes refraction. The amount of refraction depends on the wavelengths involved. Index of refraction is n equals c over v. C is the speed of light, v is the speed of light in the medium. So that's the index of refraction. Dispersion, when the various wavelengths of light separate from each other, that's dispersion. I think this is what you see in the prism, or what was on the Pink Floyd's album cover. We have Snell's law, the law of refraction. So we have dispersion versus refraction. Snell's law, the law of refraction, is called Snell's law. There is an inverse relationship between the index of refraction and the sine of the angle of refraction measured from the, so the more refraction index you have, the smaller the angle will be. I think that's what it's saying. There is total internal reflection when light cannot be refracted out of a medium and is instead reflected back inside a medium, just like here. Occurs when light moves from a medium with a higher index of refraction to a medium with a lower index of refraction with a high incident theta. So this theta is way too big, like this, or like this maybe, and it just gets reflected. We have the critical angle, the minimum incident angle at which total reflection occurs. So total critical angle is the angle that the total reflection occurs. It's the reverse sine of n2 over n1. We have lenses they refract light to form images of objects. Thin symmetrical lenses have focal points on each side. We have convex. It's the reverse, right? Concave was real and virtual. Convex has real and virtual. It's real and inverted, RI. And VU, UV like virtual and upright. And Lensmaker's equation, lenses with non-negligible thickness require the Lensmaker's equation, 1 over f 1 over f equals n minus 1 times 1 r1 or minus 1 over r2. So f equals focus, small f is focal length, and f small focal length equals half of the r, basically the center. And yeah, concave is virtual upright, diverging system, and convex lens is a converging system, like this. You can see it, concave lens like this. Perfect. We're done with in 20 minutes for this one. We have the psychology, sociology now, so let's do it. What is it above us? What chapter should we do? It's 7 and 8. So we left off from here. It's types of psychological disorders that I am actually developing right now. I'm joking. So we had schizophrenia. It has positive symptoms. It's like delusions or hallucinations. And negative symptoms are like disturbances, like avolition, you lost ambition. You have depressive disorders, major depressive disorder, and SAD, seasonal affective disorder. And major depressive disorder is at least one major depressive episode, and persistent depressive order. So let's color these with different colors. We have major, we have persistent, we have seasonal, is from winter, is easy. So the persistent depressive disorder is dysthymia for at least 2 years. That doesn't meet criteria for major depressive disorder. Now we have bipolar and related disorders. So bipolar, manic or hypomanic episodes. Bipolar 1 is at least one manic. Bipolar 2 is one hypomanic and at least one major depressive disorder. Right? So we want at least, not only one. You want one or more. Then we have, so let's color these: manic, bipolar, related, manic, hypomanic episodes. Don't need these actually, it's bipolar 1. Now we have also cyclothymic disorder, hypomanic episodes with dysthymia. We need to know what dysthymia is. Is it hysteria? Maybe. Anxiety disorders, phobias, social anxiety disorder, generalized anxiety disorder, agoraphobia. Agoraphobia is fear of spaces, I think, like lack of space. And panic disorder. And we have obsessive compulsive disorder, OCD. Obsessions are thoughts and impulses, but compulsions are tasks and impairment in a person's life. But cause impairment, repetitive tasks, but causes impairment in life. That's why it's compulsion. Then we have body dysmorphic disorder. It's unrealistic negative evaluation of one's appearance. It's like anorexia, right? Then we have PTSD. It's intrusive symptoms such as flashbacks, nightmares, avoidance symptoms, negative cognitive symptoms, and arousal symptoms. arousal for PTSD, yes. Dissociative disorders, dissociative amnesia. You can't recall past experiences. Dissociative fugue, assumption of a new identity. Dissociative identity disorder, multiple personalities. Depersonalization, derealization disorder, feeling detached from the mind and body or environment. Mind, body, environment. Then we have somatic symptom and related disorders, and they involve significant bodily symptoms. Somatic symptom disorder, somatoform disorder. A somatic symptom causes disproportionate concern. Illness, anxiety disorder, preoccupation with thoughts about having or coming down with illness. Conversion, so illness, anxiety disorder, preoccupation with thoughts about having or coming down with illness. Somatic symptom, somatoform disorder. A somatic symptom causes disproportionate concern. You should know what this is. Conversion disorder associated with prior trauma. Involves unexplained symptoms resulting in the loss of body function. Hypochondriasis, illness, anxiety disorder. One strongly believes he or she has a serious illness despite few or no symptoms. So what's the difference between illness, anxiety, preoccupation with thoughts about having? It's kind of the same thing, hypochondriasis, right? Personality disorders, patterns of inflexible maladaptive behavior that cause distress or impaired function. Cluster A is weird, B is wild, C is worried. Weird, wild, worried. And these patterns are inflexible. These people are not flexible. They are maladaptive behavior and they cause distress or impaired function. Inflexible again, maladaptive, and distress. Weird, let's color them. Weird is green. Type A, here, personality disorders. Wild is pink. Let's make it purple. And worried is yellow. Could be also red. So weird is paranoid. Schizotypal, schizoid. Wild is antisocial, borderline, histrionic, narcissistic, and worried is avoidance, dependent, and obsessive compulsive. These can be easily confused because, like, if you're paranoid, it can be obsessive compulsive. But no, it's just weird. What makes weird weird? Actually, these are good adjectives to describe something weird. If someone is paranoid, if someone is schizotypal, multiple personalities, if someone is schizoid, what is this schizoid thing? Then we have wild. These are people are antisocial, borderline personalities, what you see in teenage girls nowadays. Histrionic, history, like being crazy perhaps, like how do you describe history? And narcissistic, selfish basically, arrogant. We have worried. These people are avoidant, dependent, and obsessive compulsive. We also need to understand psychological disorders, understanding them. So we have behaviorist approach. Classical and operant conditioning shapes the disorder. Both classical and operant, so it's behaviorist. We have biomedical, takes into account only physical and medical causes. Physical and medical. Biopsychosocial, biology, psychology, society, social components. Psychodynamic approach related to Freud's psychoanalysis. DSM-5, the Diagnostic and Statistical Manual of Mental Disorders, 5th edition, categorizes mental disorders based on symptoms. We need to actually go take a look at DSM-5. I'm interested in it to see what diseases are inside it. They have biological basis, so schizophrenia. They have genetic factors, birth, trauma, marijuana use, family history. Depression increases glucocorticoids, decreases norepinephrine, serotonin, and dopamine. Glucocorticoids, are these the one that come from the adrenal cortex, like cortisol or aldosterone? Cortisol, perhaps, comes with depression and stress and stuff like that. We have bipolar disorders. They increase norepinephrine and serotonin. They are also heritable. And Alzheimer's, genetic factors, brain atrophy. These are all biological basis genetic factors, right? So bipolar, Alzheimer's, genetic factors, brain atrophy. The brain size changes. Less acetylcholine, senile plaques of beta amyloid, Parkinson's, bradykinesia, so slow movements, resting tremor, pill, rolling tremor is a different type of tremor than resting tremor, mask-like facies, cogwheel rigidity, and shuffling gait. And it comes from decreased dopamine, just like depression. So mask-like faces, cogwheel rigidity, shuffling gait, pill rolling tremor. Perfect. We're done with this chapter 2. I'm gonna take a quick break and then do the flashcards for biology. Cheers. Peace.